of cardiomyopathy. Dr. Dhandapany, please.
Hi all. Thanks, Dr. Bahal. And at the outset, I thank Dr. Kartha and Dr. Harikrishna for the wonderful opportunity. So today I'm going to talk about some of my lab work on the topic of genetics of cardiomyopathy. And I'm Dhandapany from Institute for Stem Cell Science and Regenerative Medicine, Bangalore. So as you all know, hypertrophic cardiomyopathy or a group of heterogeneous heart muscle disease leading to thickening of the heart muscle, ultimately leading to diastolic and systolic dysfunction compared to the normal adult individual as shown here. One out of every 250 to 400 people are affected with these conditions, and the causes are mutations in the sarcomeric and signaling genes. However, approximately 40% of the cases causes are still not known, and the South Asian populations are not explored promptly. So why HCM gene discovery? As you can see, the current HCM therapies rely on the type of the gene shown as in the chart. However, in many of the HCM patients, approximately 40%, as I mentioned, the genes are not known. So a comprehensive understanding of the genes are important. And importantly, the Indian HCM genetic studies are underrepresented. So the focus of my lab is to identify new genes, key mechanism, and therapeutic targets for hypertrophic cardiomyopathy. And the approach that we take is a patient-partnered clinical and basic sign approach, wherein we collect a large number of family samples of the patients and perform next-generation sequencing. Here in this case, whole exome analysis to identify novel gene variants. And once the gene variant is established, we actually check those gene variants for functional relevancies in cellular models. And once that functional relevancies are established, then we search those gene variants in a larger cohort for the specificity and the frequency. And ultimately, for a few of the genes, we actually model them using animal models for better understanding of therapeutic targets and mechanisms. So based on the focus, we have divided the team into genetics, mechanistic, and therapeutic team. And these are the current members in my team. So, as you all know, HCM is conventionally defined as a disease of heart muscle in the absence of secondary causes. However, the recent genetics data, which includes data from the Michael Arrett lab, shows that approximately 10% of the HCMs do coexist with diabetes. So far, there is no shared genetic component known for this particular conditions. So in order to know whether there is any shared genetic component between this hypertrophic cardiomyopathy and the diabetes, we actually started with the cohort 1 with a clinically reanalyzing the samples around 300 HCM patients by measuring their glycosylated hemoglobin levels. As you can see, out of the 300 patients, we actually got approximately 44 patients with hypertrophic cardiomyopathy and diabetes, which roughly accounts for 15%. So we invited all the 44 patients along with the 80 available respective healthy controls, familial controls, and we isolated the DNA and performed the exome analysis. So for the exome analysis, we have an in-house whole exome pipeline as shown here. So once we got the sequenced, so we obtained the FASTQ files and the data are filtered using Trimatic, and we compare those genome to the referral genome, GRCH38, and we remove the duplicates, PCR duplicates using Picard tool, and the variant calling is done by CATK. And we annotate those variants using ANOVAR, and the variant filtering are done by 3 methods. Either we put them into the category of in silico deleterious missense mutations along with indels. So by using polyphen score, SIFT or CAD, and we compare those gene mutations to the available public databases, which include EXACT, Genome AD, 1000 Genome, and an in-house healthy aging individuals called IDANS database. So we then take those gene variants and actually compare those gene to the mouse knockouts showing cardiovascular phenotype to arrive the point of suggesting that it might be a novel gene. So by adopting this method, we actually found an adiponectin R1 mutation in a family shown here. So these are the filters that we used, and ultimately we arrived to a particular mutation, adiponectin receptor R1, where the leucine is converted to at 157th position to histidine. So this mutation is present only in the patient, and the patient shows hypertrophic cardiomyopathy along with the diabetics, but none of the family members are having this particular variance, and most of them are healthy individuals, except for the elder brother who's hypertensive in nature. So then we looked into a second cohort with an independent cohort with HCM with the diabetes. And here in this case, we identified two more mutations in adiponectin receptor R1 in two individual patients with the following amino acid as shown here, where valine is converted to methionine at 146th position. We then screened the cohort 3, where we took only the HCM patients without diabetes, and we found another mutation in adiponectin receptor R1, where phenylalanine is at 145th position is converted to isoleucine. So we then took a cohort 4, where we screened only the diabetics patients without hypertrophic cardiomyopathy, but we didn't observe this particular variance. We then analyzed cohort 6, 5, and 6, where we took a secondary hypertrophic cardiomyopathies and dilated cardiomyopathies. However, we didn't observe this particular gene variance. Shown here are the allele frequency of the adiponectin receptor R1 in different world populations. As shown here, most of these mutations are novel in nature, except for one particular mutation, valine 146 to methionine, where it is ultra rare in nature. And importantly, these variants are not present in the Indian specific data sets, which is a Genomacia data set, which is recently released in our in-house data set for healthy aging individuals who are above the age of 80, and a South Asian healthy controls from our lab, which comprise approximately 1,800 individuals. So shown here are different family members with the respective adiponectin receptor R1 variants with and without hypertrophic cardiomyopathy and diabetes. So as you can see, all these gene amino acid changes actually convert a revolutionary conserved amino acids, and they're predicted to be pathological using various in silico software analysis, which includes SIFT, Polyphen, Panther, Metadome, mutations taster, PMUD, and CAD. And this indeed suggests that the adiponectin can cause hypertrophic cardiomyopathy. So adiponectin receptor R1 are the receptors for a hormone called adiponectin, and they play an important role in the heart, especially a cardiac protective role, through the signaling component called AMPK. So they also parallelly phosphorylate the insulin signaling cascades and thereby participating in glucose and fatty acid metabolism. So in order to know whether the adiponectin mutations can induce cardiac hypertrophy, we express those mutants in the cardiomyocyte. As shown here, expression of those mutations in the cardiomyocyte actually shows an increase in the cell size area in the mutants compared to the wild type controls. And this is also true for the hypertrophic markers, which shows increase in the hypertrophic markers such as ANF, BNP, and beta-myosin heavy chain. This data indeed suggests that the adiponectin mutation can induce cardiac hypertrophy. So in order to know the mechanism behind it, we took the representative mutation, the V146 and phenyl 145I. So the interesting thing is like both of the set of these mutations, we took the representation, one as a HCM with the diabetes and one as a HCM alone. Interestingly, both of these mutations doesn't follow the conventional AMPK pathway, whereas the hypertrophic cardiomyopathy with the diabetes took an unconventional pathway, P38 mTOR pathway. And interestingly, the hypertrophic alone causing mutation actually took the ERK pathway. However, they didn't activate the mTOR P38 pathway. So this indeed suggests that the adiponectin variants actually activate diverse pathways to implicate its function. So in order to know whether this mutant affects the glucose metabolism, we actually took a representative mutants and expressed on the cardiomyocyte as shown here. So the mutants actually shows an impaired glucose metabolism compared to the wild type and beta-gal expressing cells. And they also shows an upregulation of the serine insulin receptor as shown here, which is paralleled with the downregulation of AKT and phosphofructokinase. And these data indeed suggest that the adiponectin mutation can affect glucose metabolism. So in order to know whether this happens in an in vivo setup, we actually generated a mouse model carrying a representative mutations V146M. And as shown here after the treatment of the Maxwin. So this particular mouse model actually shows heart failure. They first show hypertrophy and they quickly progresses toward the heart failure as shown in the ultrasound image over here. So the mutant mice also shows and degrees fractional shortening, and also their cross-sectional area shows an increase in the hypertrophic of the cells size as shown here. And the mutant also shows an increase in phosphorylation of the phospholambin and a decrease in SRCA2A, which indeed suggests there is an abnormality in the calcium signaling. The mutants also shows increase in the hypertrophic markers, which include ANF and beta MHC. And all these changes we are able to rescue by means of an mTOR inhibitor rapamycin. As shown here, the rapamycin is able to rescue the phenotype of the mutant mice, and they can able to bring back the fractional shortening, and also the cross-sectional areas are reduced, and they're also able to restore the phospholambin SRCA2A and also the hypertrophic markers. So the mutants are able, the rapamycin are able to rescue the fibrosis that are shown in this mutant mice, as shown here, and the mutant all mice are able to rescued by the rapamycin, especially the signaling cascade mTOR, as shown here. The mTOR is signalings are able to get back to the normal, along with the downstream signaling cascade 76 kinase. This indeed suggests that the rapamycin can rescue the adiponectin mutations, and especially the adiponectin receptor R1 can cause hypertrophy. So in summary, the adiponectin mutations actually causes hypertrophic, and depending on the positions, basically they are either activate P38 mTOR or ERK. And if they are able to activate the P38 mTOR pathway, those hypertrophic cardiomyopathy patients are associated with the diabetes. And if they are activating only the ERK, then they are activating only hypertrophic cardiomyopathy. So with this, I stop and I take any questions.
Thank you, Dhandapany. That was a wonderful presentation, and you've been able to show the whole pathway. I just have one question: that is this? Do you have any heart tissue in this? Because is it true hypertrophic? Does it resemble the sarcomeric mutation type of hypertrophic, or is it a type of a phenocopy?
So yeah, that is really a good question. So, I understand the point that you're coming from. You're asking the question whether it's a sarcomeric kind of, or it's a glycogen storage disorder, because since the AMPK pathway is involved in this process. Yes, we do have a heart tissue. What we didn't observe is there is no infiltration of any of these components in the heart tissue, whether it resembles the sarcomeric kind of hypertrophy. And then we looked into any of the sarcomeric polymorphisms are present in this particular patient. However, we didn't observe any of the polymorphism in this patient, except for the one patient where we do observe a beta-myosin heavy chain polymorphism.
Thank you very much. If there's no other question, then thank you, Dhandapany. That was a wonderful talk, and you dissected out the whole pathway.
Thank you very much. Thank you very much.